Charlie, I think ConferZoom will still record it. Like, I think it'll show up in our past lectures in ConferZoom.
Oh, you're right. Okay, so I'll send out a little message. I'll view it first, and then I'll send out a message that the entire one is there. So we may be recording for nothing right now, because you're right, ConferZoom records it as well. They don't want us to use certain recording methods because it takes up all of their storage space, and I can understand that. People record stuff 5 years ago. They still got it on there. Nobody wants it. Okay, you can see me, right?
Yes.
Okay. And here's the test that I was talking about. Somebody else was asking about it too. So I'm going to go through this test. And then when we get to the portion where we are looking at the pancreas and the liver and the stomach and the small intestine that you asked about, we'll slow down, okay? And this first one is, like I said, it was a previous test, wasn't given on Canvas. It's not made for Canvas, obviously. The organ that the slide's taken from is the kidney. Okay. Now I can sit here and ask you questions about this all day long, or cover more ground if I just go through it. What do you want to do?
Just go through.
You can go through it first.
Okay, we'll go through it. These are Bowman's capsules. Bowman's capsule has within it a glomerulus. Okay, and surrounding are all the kidney tubules. What you need to know, first off on this slide, what organ are you looking at? Okay, this this can't be the pancreas. This can't be the pancreas. It doesn't have tubules running through it. The pancreas has islets of Langerhans, but they're not large like this with big gaps around them. Recognize these as Bowman's capsule and glomerulus, and you know that it has to be the kidney. Lungs, this doesn't look like sponge. Ovary, oh my God. Ovary of what animal? Are these follicles? No. If they were, there's going to be a whole bunch of these animals pretty soon. Okay. And the liver, it doesn't have anything at all relating to the liver. So in any event, the organ is the kidney. What's the structure at the tip of the pointer? Now, you're not going to have to worry about this, but the tip of the pointer, I would immediately tell everybody that the tip of the pointer is touching this structure. That's where the tip is. Okay, it's not out here. So this is the glomerulus. Always look, if you get a question like this, for agreement between kidney, in this case, and glomerulus. Where are you going to get the agreement or disagreement on a Canvas test, where there's only one question per picture? Picture is going to be shown twice. First time, name it. Second time, what's the structure? Okay, so we can still do it. Now this is the liver. Okay. This is a liver picture for you. You see the tiles. It's supposed to be six-sided, but that's, textbook. They all each have a central vein, central vein, central vein. hang on. When you're looking for any of these things on your own, I want to see the histology of the liver. You got to get the word histology in there. You won't get anything you want. And then hit images. Okay, this is liver. You see the tablets. The color means nothing. Okay. This is a great shot right here. Central vein, central vein, central vein. They all have a central vein. Every lobule has a central vein. But you're not going to be able to see it unless the cut goes right through it. And it didn't. Liver. I'm only picking those that I think I could possibly use. It's too bad they put in this. I don't know what this is in here for. It's like it's sewn together or something. Because otherwise, that's a great picture of liver right there. Okay, this is definitely liver here. This is the central vein. This vein has blood cells in it. That's why it's colored. And over here. And if we can count, hopefully get 1, 2, 3, 4, 5, and the short sixth one. Wherever 3 cells come together, you have a triad. 3, 1, 2, 3, a triad. 1, 2, 3, a triad. Remember what's in a triad: an hepatic artery, a portal vein, and a bile duct. Okay. Now, you wanted to see that with reference to the pancreas. So I'll move ahead. Here's pancreas. Okay. Now, you just saw liver, and this doesn't look anything like it. There's no particular central vein. There's no tablet-looking effect that you have here. It's all rather homogeneous, except for the fact that it's got embedded these structures. And these are islets of Langerhans. Islets of Langerhans. That little teeny thing is an islet of Langerhans, but it's like if this were the surface of a dirty ocean, that's been pushed down. It's mostly below the surface. I'm only worried about these. These are islets of Langerhans. Question, what organ? So the answer here is pancreas. Capitalized for no particular reason. Structures at the end of the pointers. These are both islets of Langerhans. The small little cells that you see in here are alpha and beta cells. Alpha cells give rise to glucagon. Beta cells give rise to the insulin. And then I'm giving you a little more than you asked for. The area around here, darker cells, the and denser area, these are acinar cells. Okay? Pancreatic acinar cells, if you want to be precise, and they produce pancreatic amylase, which is delivered by ducts through the pancreatic duct to the ampulla vater and the duodenum. So we have a digestive gland out in here, digestive function, and an endocrine function both. Do you think you can tell the difference between the two? There's the other one. I think so. Okay, I'm going to keep on going in
Charlie?
order. Yep.
So are all these in order with the ones, the photos that are in your, the Charlie site?
No,
No, they're not in order? Okay.
they're not in that order. This is a test. This is an actual test. Okay. No, nobody would put it in the same order. They're not, but the ones that are on the Charlie site are all things that you should be able to identify. And a lot of the pictures on the Charlie site are used here. As I come past one, I'll mention that. Now I'm going to go still in the same order until we hit the small intestine and stomach, and then we'll compare those. Okay, here's hyaline cartilage. Now, somebody said that they were having some difficulty distinguishing hyaline cartilage from what else? I guess that person's not still here. Looks like everybody that was here is still here.
No, I'm here. It's hyaline cartilage from, it's actually just one picture where I find it hard to distinguish, but from simple squamous.
Okay. Well, this is not squamous for one thing. Squamous has to be flat. And these individual areas here are chondrocytes, okay, inside of a lacuna. Actually, the lacunas have coalesced in this one. They always look to me like they're floating in plastic, but you may think of it as a milky substance, although the color can be anything. Okay. What kind of tissue is this? We are looking for hyaline cartilage. And where in the body would you find this tissue? Tendons? Oh, you wouldn't want cartilage in your tendons. You don't want cartilage in your salivary glands. You don't want cartilage of this type in the epiglottis. In the epiglottis, you want elastic cartilage. It's articular cartilage at the end of the long bones, not inside of them. This would be the answer here for protection. Pretty slide. It's got hairs on it. And although there are other types of slides or cells with hairs, nothing that we've done with hairs is anything ever but pseudostratified ciliated columnar epithelium. The word ciliated's been left out here. Doesn't matter, it's the same thing. Here's the cilia, pseudostratified because it looks like there's many layers here, but there's not. Every one of these nuclei is in a cell attached to the basement membrane. Where do we find this? This type of epithelium is common in the respiratory tract. So that's not it. That's not it. That's possibly it. But this is really it, the trachea. Okay. I said the lung was possibly it because it's part of the respiratory tract, but this tissue does not exist in the lungs themselves. Okay. This is meant to beat out dust and other particles and drive it upwards so that it can be swallowed, actually. If those particles get down into the lungs, it's too late for that. Here's a salivary gland. You see the lobes. We saw lobes in the liver. We did not, okay, and These are salivary gland, as I said. These are acinar cells, dark ones, secreting salivary amylase. There are ducts, as in any exocrine gland. This question simply says, what are you looking at? Okay, let's look at it and say salivary gland. That's a maybe. Testes, doesn't look anything like that. Liver, we just looked at, doesn't look anything like this. Large intestine, the one that looks like an octopus. No way. Thyroid, oh, well, you probably tell Charlie that we didn't do thyroid. That's true. And that's not the answer. It's salivary gland. Now, there are a couple of tissues that don't lend themselves easily to memorization. So instead, get familiarization. In other words, you see it enough times, you know what it is. Just like a person, you ask them their name a couple of times. Okay. I'm really bad at names. I mean, I can go into a class that has 40 people and go around the room and ask everybody their name one time, taking a roll. And then I can challenge the class that I know the first and last name of everybody in the room. And they'll go, oh no. And I say, well, who wants to bet their first 10 points against me? And they always lose because I can do that instant recall stuff. Come the next day, I don't remember any of them. Okay. But you can do that here just by remember the name of it. Next time you see it, same thing, and pretty soon it gets tiring. Okay, this one should not give anybody trouble, or you are going to have trouble on the test. This is the skeletal muscle. Striations, nuclei on the periphery, areolar connective tissue. Beautiful, beautiful one. So many spider webs and flies caught in it. This is the neutrophil, otherwise known as the multipolar or multilobular. Okay, I just muted one of you. Okay, the blood type below is then neutrophil. This is lung. Lung looks like a shattered windshield when it hasn't broken out yet. Okay, it also looks like sponge. You got to remember that the simple squamous is a predominant feature in the lung. These very, very thin outlines to the alveolus. The alveolus is the smallest of the chambers, and they're all over the place in here. A neuron probably won't give anybody any trouble. Glial cells around it. This is seminiferous tubules. This one often gives people trouble, but when you look at it, see the lumen, that's obvious. Okay, as long as you don't think these are globular and you realize that they are cylindrical. Okay, then you look at the periphery and you don't see a distinct type of cell inside of there. Just cells dividing and coming in. Okay, this one has stratified squamous epithelium, and that's from here to the top. That's the epidermis. The cells in the stratum basal, and spinosum, and granulosum, these are those cells here. Are larger, but as they're moved closer to the periphery, they flatten out. They're very flat in here. Flatter even still. A generalized function of this tissue, protection from abrasion. The eye, choroid coat here, sclera back here, doesn't have to be white, and then a big retina right here. Always start with the darkest part, you can't mistake that. That's the choroid coat. Okay, this one is the small intestine. The small intestine has finger-like villi. If you ever took in your science class anything to do with worms, you might recognize this as a planarian. If you don't know what a planaria is, forget I even said it. It's not necessary. These finger-like projections are the villi. In between villi, way down here, we have what are called intestinal crypts. Intestinal crypts. The villi is a projection upward, outward. Okay, they don't have to fit together like they were sliced apart. They don't. They have on their outside the microvilli, which are simple columnar epithelium. This is nice because it also shows some of these clear cells here, and here, and here, and here, and over here. These are goblet cells that are common throughout the digestive system. They secrete mucus for the movement of the contents. Okay, so first thing to recognize, I never had any trouble with this one, and a lot of people don't have any trouble with this one, but there's, for each person here, there's a pair of slides that gives everybody trouble, or gives you trouble. But you notice that they don't fit together. They don't all fit together. This one's got difference here. Okay.
Charlie, I'm sorry, can you repeat answer for number 15? You said it was neuron, but I don't see that answer on there.
Oh, it's spinal cord.
Oh, it's fine. Okay.
Yeah, it's the only thing that has to do with the nervous system.
Okay, thank you. Charlie,
Yes.
about that one as well, is that what the tissue of the neurophysis would look like as well?
Yes.
Okay,
Yes.
thank you.
Okay, now I want to move forward and see if I got a picture of stomach here. I don't have a picture of stomach here. Okay, so I need to get one quickly. Okay, this is stomach. Okay, And these things are not villi, because between these two structures, it seems, there's a slit, and these slits fit perfectly together, leaving a channel. These channels go down to here, down to here, down to here. The channels lead not to intestinal crypts, but to gastric pits. I'm throwing out some words that I haven't used before, and they won't be on the exam. This is to clarify something here. The fingers that you saw a moment ago, you had long fingers, short fingers, and here they're all exactly alike. This is an artifact. In other words, just an accident in making the slide. All the way across, they're flat, even. That's because there are no villi in the stomach at all. These are not individual fingers. These are slits downward. So imagine for a second that this was a bunch of brownies or something, or a cake. Brownies are already slit all the way across. Now take a very sharp knife, cut, cut, cut. Okay, there are no projections being formed, only creases made between. At the bottom of the slide is smooth muscle.
That helped a lot. Thank you, Charlie.
Okay. Okay, that's where we were. Here's where we are. This is a follicle. This is an ovum. What's the question? The organ is, what organ is this tissue taken from? Ovary. And what's the structure at the end of the pointer? A follicle. Here's our picture of the pancreas that we took time with just a moment ago. So it's taken from the pancreas, and it's pointing at islets. Actually, I prefer islets of Langerhans. I must not have made this question up. What type of tissue is this? The color fools people. Forget that there's any color. It can be any color in the world. It's the parallel movement of the striations that you see. And yes, you see some wavering here, but the wavering is exactly the same in the striation next to it. The wavering is because a knife has cut across the tissue. It's called microtome. And it must have wavered a bit or not been the sharpest knife in the world. You've all seen this wavering when you have used a knife on butter. Okay? Sometimes it's straight, sometimes it's straighter, sometimes it's wavering. This is all tendon, always going in the same direction. What type of tissue is tendon? Dense, regular connective tissue. The tissue seen at the end of the pointer. I don't care that there are tubes, chambers, or anything else. I look at one of those cells and it's cuboid and it's a single layer. It's simple cuboidal. There can't be any mistake in that. Where in the body would we find them? Oh, well, then I have to realize, look at how big, long this tubule is. Where did we find simple cuboidal? The only place was the kidney. Here's the one I called an octopus. I like this slide better than some of the other slides that are posted for you for colon. Okay, name the organ, colon. It could also have said large intestine. It always looks to me like a bunch of tentacles coming over a wall, but I'm thinking in 3 dimensions, and this is a 2-dimensional slide. Okay. The epidermal layer is here. The epithelial layer, I should have said, here. And since it's part of the intestine, I'm sorry, the GI tract, these must be simple columnar epithelial cells, and they are. But now we've progressed more distally or farther along the GI tract, and there are more goblet cells, more of these white cells. And they give the sucker-like appearance of the so-called tentacles that I see. Okay, so it's colon, and the epithelial layer is simple columnar. This is the tongue. What's being pointed at are taste buds. Okay, what would you like to see now?
I think what you mentioned earlier about the roads in the uh, in the nervous system, I think helpful.
Okay, the nervous system is up here. Respiratory, that'll be on there. There are the first part of the test that I looked at the other night, of course, again, changed some things, but the respiratory system's the very first part. And I don't know that the third lecture is the one to be starting with, but I'm going to. Okay. 2 major parts of the nervous system, the central nervous system, brain, and spinal cord, and all of the rest of it is the peripheral nervous system. The incoming stimuli travel on peripheral nerves to reach the central nervous system, and once leaving, the motor neurons also travel on peripheral nerves. Okay. The central nervous system, you might think is more static in place in the spinal canal, but the other nerves, although they're more numerous outside, they pretty much have their place too. So to line them up, here are our spinal nerves, as they're called. The spinal nerves are the go-between. You have peripheral nerves coming in to the spinal nerve. The spinal nerve is also a peripheral nerve. Okay, it's just a different type. Okay, that sort of is the exit or entrance into the vertebral column. There are 8 pairs of spinal nerves in the cervical vertebra. Don't try to count them here because you can't see them all. But there are 8 pairs. Here's our vertebral artery we saw before. 8 pairs here. In the thoracic region, we can count 12 vertebrae, and underneath each vertebra is a set of spinal nerves. There are 12. 5 lumbar, 5 sacral, and one not shown is one single coccygeal spinal nerve. These yellow things that are sticking out in all of these cases are the spinal nerve. They're small. They're the go-between between the peripheral nervous system and the central nervous system. But they have to be one of the or the other themselves, and they are all peripheral. The spinal nerves are all mixed. All of these yellow structures are mixed. That means you have afferent pathways and efferent pathways in and out. The total number of spinal nerves is 31 pairs. 8 pairs here, 12 pairs here for 20, 5 and 5 for 30, and that one coccygeal for 31. The spinal nerve, the only thing I want to show you here is just this little piece here. This is what was in yellow just a moment ago. Okay. The nerves go in through the dorsal side, the afferent nerves into the dorsal side, the efferent come out the ventral side. But both pass through the spinal nerve. You must be very familiar with this diagram. There are many questions on the test asking you to name the parts. So what are the parts? Well, here are the spinal cord itself. And in the horns, we have the dorsal horn, which receives afferent input, sensory input, same thing. Down here is the ventral horn, which produces efferent output, motor output. And the bulge is a lateral intermediate mass. That'll be the start of our preganglionic neuron when we get there. We're not there yet. Let's finish naming this guy. Here's the ventral root. The swelling is always the dorsal root ganglion, and the dorsal root here always brings afferent or sensory impulses. The afferent and efferent meet and share the spinal nerve. The spinal nerve is short, so short, in fact, that most of this one is hidden in my drawing behind this portion here of the chain ganglia. I say it would run from here to here. Coming this way, motor-wise or efferently, when you leave, we have two connections to a chain ganglion, also known as a sympathetic chain ganglion or a sympathetic trunk ganglion. You have one way called the white remus, which is the most lateral. It's the way in. You have a gray remus, which is the more medial of the two. And the gray ramus is always on the way out. What happens between in and out? We said it last week, but I haven't said it yet today, and I will. In and out of this structure, the chain ganglion or sympathetic chain ganglion. You do not need to know the collateral ganglion. What we have here is the setup of different pathways. I'm sure if you design city streets, you could do better than this, but this is the way the body works. You have incoming from the body itself, the sensations being brought in by pseudo-unipolar neurons. The pseudo-unipolar neurons make the dorsal root ganglion by the accumulation of their cell bodies all in one spot. And then running away from the cell body is the axon itself, much thinner. So we have the way in, and then we have the way out. Now the way out could simply be to turn around and go to a muscle. You would still be part then of the voluntary nervous system. You might have a sensation coming in, you do, but is that going to activate the voluntary or the involuntary nervous system, or both? Well, when it activates the brain to cause the formation of an impulse to go out to a muscle, it's going to the voluntary nervous system. By the muscle, I meant skeletal muscle. So this could be very simple. In with the information, out with the movement to make a correction. It's also possible that the brain decides not to do anything at all about something. When you see something, you see something. All day we do this. We see something, and that's incoming information. Goes to our brain. Do we necessarily do anything about it? Care about it? Think about it? No. Okay. So it would be great if this was just in and out along the voluntary nervous system, but it's not. You also have the involuntary nervous system, and it's going to use the same incoming information here, and also incoming information from the body when your stomach aches or something is wrong internally, and you may not feel it. It could be pH level, it could be your blood pressure, it could be a lot of things. But that information is set also by the visceral sensory system. That you might want to refer to that first chart. I'm going to go over it irregardless tomorrow. Okay, but the incoming information can also then be sent to the involuntary nervous system, which we also know is the autonomic nervous system. It's going to use the same path. Why make another road? Okay, we use the same path here. But now, instead of moving on down to a skeletal muscle, it's going to, in this scenario here, in the sympathetic system, it's going to move into the sympathetic chain ganglion through a white ramus. From there, it can move up or down and synapse with the next neuron, and then travel to the organ that has to be told what to do or be innervated. When it travels out of the chain ganglion, it always exits by the gray ramus. I want to get to the next picture. Okay. This is going to be following the sympathetic pathway. That pathway is initiated, though, by incoming information. Now we have outgoing information. Okay. Motor neurons are activated. But these aren't going to the skeletal muscle. Instead, they're going to go to the smooth muscle or glandular epithelium. Okay. And you have two neurons in the pathway to get to an organ or a glandular epithelium, which is usually a gland. The first neuron is the preganglionic neuron, in this case, of the sympathetic system. Okay, how do we know it's sympathetic? One, it's got a white ramus. Only the thoracolumbar vertebra have the white ramus, and the thoracolumbar vertebra constitute the thoracolumbar system or the sympathetic nervous system. So since there's one of these here, then this is going to be a preganglionic sympathetic neuron, comes into the ganglion. It may do the same over here and continue on without synapsing yet to another ganglion. And remember, since this can fray, it's a nerve. It has many neurons, can go up, down, down farther. Well, then one command coming along can suddenly spray out to lots of different ganglia and reach different parts of the body. This shows two ganglia, the preganglionic neuron here. It enters into the white ramus, and this one immediately synapses, goes up, and then back out. And now it's going to go to the organ. Is there a reason? Is there a reason, can you think of it, that it has to even synapse? Why not just go right to the organ? Why not just go right to the organ? Why bother synapsing?
To connect to the postganglionic nerve?
It does. That's what the result is. Yes, that's the result. But why have two neurons anyway? Why not just go straight to the organ?
Can the preganglionic neuron synapse with multiple neurons to send the signal to a lot of different places at once?
Yeah. Who was that?
It was me, Jenna.
Okay, Jenna. That's exactly right. If you only had one neuron, you have no combination of effects. By having a synapse here, or one down farther, the combinations are endless that you could have. You could be stimulating several organs. There's not just one organ in the digestive tract, for instance. It could be several. But you want some at this level. So if you came in here, you better synapse here as well so that you can have influence at this level, as well as this level, as well as the level above. Okay. So the fact that it does synapse here is a necessary consequence of the fact that we're trying to stimulate several areas at once. Well, did I put it there? In the area of the sacrum, the spinal cord initiates the preganglionic neuron. It's a motor neuron, but there's no white remus and there's no way to get into the party here. So you must go on. It gets the next longest neuron, which is the splanchnic nerve, which goes right to the organ. Okay. So unlike what we just said with the sympathetic system branching and moving up and down and at several levels, in the parasympathetic system, it can only work at the level that produces it. That's not to say that the splanchnic nerve won't subdivide and go up to the stomach and down to the colon. No, it will. Okay, but it's not going to be a fast pace like the sympathetic nervous system is able to do. The sympathetic nervous system can get its preganglionic neuron all the way down here and farther and farther. And then it would synapse with the postganglionic neuron. But in moving downward like that, here it comes downward and it enters this sympathetic trunk ganglion that I've drawn here from above. But there's no way in, not from here. There's just a way out to here. So a sympathetic trunk ganglion can generate a sympathetic postganglionic neuron to take and travel out, or even come down the splanchnic nerve and take over if need be in the case of fight or flight. So when things need to happen fast, it'll be the sympathetic trunk gets the job done. These are the last models that we did, and I just wanted to get one group of them out of the way so I don't have to do it tomorrow and we have more time for other things. So what I'm going to do is go through these, and I mean fast. You stop me if you need it slower, okay, or when you need it slower. But you need to be familiar with the following things. On the ovary, first off, that's what this is. You need to know the follicle, the corpus luteum, and the corpus albicans. You need to know the secretion of the corpus luteum, progesterone, okay, and that will probably do it, except also, of course, know that the ovary is an endocrine gland and a body organ, reproductive organ. A ureter attached to the kidney. Not a good picture. I've got better. Know every part of this. Every part of the epiglottis. Be sure to know it's elastic cartilage. Here's the arytenoid cartilage, which is hyaline, cricoid, and the cricoid on this side, and the thyroid are all hyaline cartilage. C-rings are cartilage. Thyroid is an endocrine gland, secretes calcitonin and thyroid hormone. Parathyroids secrete parathyroid hormone, both involved in the deposition of calcium into bone. The mesocolon is a great shot for that. Okay. The spleen, know that it's a reservoir for some blood, and also it's the largest lymph node in the largest organ in the lymphatic system. The spleen is also responsible for breaking down red blood cells. A Ostrom, Tinea coli for sure. On this model, know the ovary. Do not worry about the innards of this ovary. The larger model we just had will do that. Here's the ovary. Here's the os fimbriae. Okay. I've looked at the test, and when I've used os fimbriae in parentheses, I have infundibulum, so there'll be no confusion. Here's the oviduct. I will only call it the oviduct. I'm not going to call it fallopian tube or anything else. No uterine horn. No. Prostate gland, its function to produce alkalizing fluid to add to the semen. Okay. That's an exocrine gland. That's not an endocrine gland. Same thing here. There's no difference with that one. The adrenal gland, composed of glandular epithelium. Adrenal cortex produces steroids. Adrenal medulla produces epinephrine. Okay. Influenced by ACTH. Adrenocorticotropic hormone. That only affects the outer portion, does not affect the inner portion. Bladder and ureter, no problem. Another ureter shot, better shot. Here's the baby picture for Mike, the thymus, right here. Maturation of T cells, invaluable in forming the immune response. This one is showing the spleen, but it's a great picture at the same time. No, it's not a great picture at the same time. I was going to say of the diaphragm, but it barely shows. It's covered by the lung above it. Mesentery proper or serous membrane, holding together the parts of the small intestine. Here's the greater omentum. You have to sort of trace this backwards carefully, but you're going out. So this is the ascending loop of Henle, distal convoluted tubule, collecting duct. Here's the cortical radiate, also known as the interlobular. I believe I have on the test, I put it in parentheses. I think I call it cortical radiate. I prefer that, but I put interlobular in parentheses in case that's what your book had. The arcuates, can't forget those. This model has beautiful renal pyramids, a nice lobar artery, of course, nice ureter, renal pelvis, where two come together. We have a major calyx where there's just one. We have a minor calyx. Greater omentum showing here. On this model, obviously that's liver. Know that that's spleen. Know that that is the gallbladder, the exit from which or entrance into which is the cystic duct. And to or from the liver is the hepatic duct. They combine to make the common bile duct. An oil gland or sebaceous gland is a holocrine gland. You need to know the word holocrine. Sudoriferous gland is a sweat gland, secretes sweat. You need to know that it's a merocrine gland. Here's the dermis. And then starting with the dark material to the top is the epidermis. The pharyngeal tonsil. The palatine is here. And then the shot of the nephron, arcuate, art vein, and artery, cortical radiate going up, also known as an interlobular. Here's the afferent arteriole going into the glomerulus, which is surrounded by Bowman's capsule, and comes out as the efferent arteriole, and spreads out over the entire network of tubules as the vasa recta to reabsorb valuable material. From the glomerulus, the plasma is expressed in the proximal convoluted tubule, descending loop, ascending loop, and then distal convoluted tubule to the collecting duct. The farther along you go in this chain of movement to the collecting duct, the more the fluid inside is described as urine. Know that the muscle of the stomach and other internal organs are in smooth muscle. There are a few muscles, as you recall, that are internalized, but they are skeletal muscle. For instance, the diaphragm, the skeletal sedal, and the psoas major, the iliacus. But the majority of the internal muscles are smooth muscle. And don't forget the other name for smooth muscle, visceral. Okay. It may be used on the test. Okay. We went a little longer today. It didn't feel like it to me, just running around quickly like that. Maybe we got a bit done. What I need you to do, or would hope that you do, is if you go through the material, if you're studying tonight, you almost need to because we're so close to the end. The thing I'd appreciate is some questions tomorrow, or suggestions or direction as to what it is you most would benefit from. Okay, and then yes, there will be an email telling everybody that Wednesday is the last time I'm going to accept anything. And I don't mean it at you. I'm not trying to rub it in. You know it. You heard it. But there's some people that may not hear it, and I have to make every attempt to get to them. Okay. So if you have any questions, hang around. Otherwise, have a good night.
Thank you,
Thank you,
Charlie.
Charlie.
Thank you, Charlie.
Thank you, Charlie.
Thank you, Charlie.
Bye-bye.
Thank you.
You're welcome. I wonder what happened to Randy. You're the winner. So with Taryn, with all the moving around that you say you do and everything, I guess you're watching the Olympics and such.
You know, I'm actually not watching the Olympics yet. That's going to be part of my treat when I'm done with this semester, is to backtrack and watch some.
Oh, you're recording them or something?
I think you can stream them online.
Oh, probably. Yeah,
Mhm.
probably.
I'm looking forward to gymnastics and synchronized swimming.
Yeah, I've already seen those. So I won't spoil it for you.
Okay.
Yeah,
Thanks.
there was one upset, real good upset. Okay,
I think I did hear about that
okay,
in the news.
okay.
Alrighty, thanks, Charlie. Have a good night.
Night.